Hello, my name is John Butler. I am the CEO and founder of QuantumCyte. At QuantumCyte, we're building tools and workflows for the single-cell biology research community. Now, why would you want to study single cells? Well, it turns out that the human body has about 70 trillion cells in it, and each cell plays a role in maintaining the overall health of an individual. So if you want to understand human biology and the human condition, and if you want to get to the core of disease, you've got to study individual cells. Now, the field of single-cell analysis is a new and emerging field, as evidenced by the increase in NIH funding for single-cell analysis studies in just the last 4 to 5 years. We're also seeing initiatives like the Human Cell Atlas, where the goal is to map every single cell in the human body. As an emerging market, researchers are looking for new capabilities to help advance their research. They're looking for the ability to take individual cells and place them into individual containers, but not just one cell, thousands of cells. They want the ability to treat those cells with reagents and then measure a response of those individual cells. They want the ability to go into the cell and pull out the genetic material, RNA and DNA, and they want to get sufficient quantities so that the data that they get after analyzing that material gives them strong conclusions. They also want the ability to associate that genetic data with specific cells in their test. And finally, they need flexibility, the ability to modify their experimental conditions in their lab without having to wait for a company to deliver a new product that may or may not help advance their research. Now, how does QuantumCyte deliver these capabilities? Well, what we do is we put individual cells into drops on a piece of glass about the size of a microscope slide. We then take another microscope slide and deliver reagents into the same drops in the same grid pattern. We then touch the two drops in what we call a kiss transfer. Now, what happens when those two drops kiss is the cells respond to that reagent. And we measure how this cell responds and how this cell responds and how this cell responds. And we pull out, again, that genetic material, the RNA, analyze it on a standard sequencer like an Illumina or a PacBio, and take that data and figure out how those cells are responding and how the cells are responding differently. Now, if we can get this type of a product out, it's truly amazing what we can do to understand disease. We built a tool, we call it the Q1. The Q1 was designed and developed by our CTO and founder, Bidan Chowdhury. The system integrates some advanced optics from Intel that allow us to deliver the capabilities that we want to the customer. The system is housed in an environmentally controlled chamber, and the two slides I just mentioned are inserted into the system. The optics with the hardware and the software find one of the slides, they pick that slide up, and then they place it into a position directly above the receiving slide. You'd have the reagent slide and then the cell slide. The optics align these drops to ensure that we get proper and consistent delivery of reagents to those cells. And here's a movie showing the KISS transfer happening. One more thing I didn't mention is that we can actually address the cells, if you will, multiple times with different reagents. We have data showing that we can detect live and dead cells in our system, and we've shown that we can extract this RNA and measure gene expression in the live cells. We've also demonstrated that we can isolate over 65% of the total messenger RNA from the cells within our system. I'm proud to announce that in the last 4 months, we've booked $375,000 in revenue on the sale of our instruments. We have collaborations with Harvard, Stanford, as well as the Icahn School of Medicine. When we compare our technology to the current product families on the market for single-cell analysis for these capabilities, we show that the QuantumCyte technology can deliver all of these capabilities, and we have a competitive price. The market for single-cell analysis is $1.6 billion with a 20% growth rate, And our business model is to sell instruments and consumables to the research market, and we'll be using third-party manufacturers to manufacture the product. Lifetime value of our products is about $640,000. We have 6 patents pending with priority dates of some of the patents back to September 2012. We've got a full-time, commercially focused team with over 100 years of experience in high-tech, biotech, as well as investment. And we have a world-class science and business advisory board. Dr. Ron Davis from Stanford's on our board, Dr. Anders Grinnet Jepsen, CTO of Intel, Dr. Chris Love from MIT. Dr. Chris Love is known for his expertise in the field of single-cell analysis. Dr. Carl Navina from Dana-Farber, Harvard, and Dr. Brock Siegel from Bioinflection Point Partners. He's not only a business advisor, but he is an investor. In the last quarter, we've booked 2 orders, and we'll be installing our first unit at Dana-Farber to start running clinical samples in October. We'll realize our first revenue in Q1 of 2018. At the end of Q1 of 2019, we'll be in production. We're currently raising $4 million. We have $1 million committed. My name is John Butler. I am the CEO and founder of QuantumCyte. If you're interested in joining us in this journey, please contact me. Thank you.